So what we did was put together a brochure, and we mailed this out to a series of different agencies and people who are working in the field, and it turned out to be marvelously successful. We had so many collections coming in. It was really wonderful to be able to add all of these in to the project. A lot of them we are still working on, but it's nonetheless pretty exciting, and we're very grateful for all these samples that were donated to the study. Okay, really briefly, I'll go through some of the results that we got. I'm going to start off with the first marker that I described. This is the universally amplifying plastid marker that I described at the beginning. We have about 76 sequences for this right now. This marker proved to be reliably amplifiable and sequenceable for several of the groups, including the Alvales, or these flat blades, and the Bryopsidales, and several of these other groups here, Chaetophorales, Agnometales, and Charales. So reasonably good across a whole series of different lineages. We had a lot of problems with this group here, the Cladophorales, and there are a fair number of species in this group. So it's a little bit problematic that we weren't able to get some really good sequences for that group. A high proportion of these sequences were, not these sequences, but others that we generated, were contaminants. And this is sort of an ongoing problem with this marker. We're constantly troubleshooting it, trying to tweak our protocols so that we end up with fewer contaminants. Haven't exactly figured out how to get around that 100%, but we did redesign the primers slightly for the green algae, and it got rid of about 60% of those problems. So we're making progress, at least on that front. It seems to be a trade-off of the universality of these primers. These are only a couple of nucleotides different from even the sequences that are homologous in bacteria. So it's tricky to be able to always hone in on just a very specific group. These are primer sequences that are present in pretty much everything that has a plastid plus cyanobacteria. But one of the things it did do was highlight a lot of taxa that are in need of systematic revision, and I'll show you one example. This is the genus Codium. We have a number of representatives here in the Hawaiian Islands. And you can see here that just looking at our really simple neighbor joining trees, these are not bootstrapped, they're just barcode-like analyses. You can see that Codium edule is broken up into a couple of different groups, and you can see that we have Codium reedii right here in the middle. So this is just one example of a group we'd like to go back to and look at in a little more detail in terms of the morphology and try to determine what is going on. Okay, secondly, we looked at this RBCL gene. This is the large subunit of this gene. We only generated about 22 sequences for this. The primers were not broad enough to be able to amplify across this range of green algae that we were looking at. We were able to reliably amplify and sequence the Alvales and some of the Bryopsidales, but not a lot of the other groups. This, in combination with the fact that we had much better results from the previous plastid marker, meant that we didn't fully pursue it. We didn't go a lot further than this. We didn't see that this marker would add a whole lot beyond what we were able to get with the UPA. Nuclear LSU results. We obtained about 34 sequences for a broad range of Hawaiian green algae. Right here I'm going to exclude the Alvales and show them to you separately on the next slide. Now this is the only marker that really reliably worked for this group here, the Cladophorales, and also the genus Halimeda and the genus Caulerpa. Now for that reason, we think this is a pretty important marker for research, and we're definitely recommending this as one of the two that we continue to retain and use in our assessments. I'd also like to note that Hawaiian Caulerpa is being studied in a lot more detail as a result of some of the things that we're finding with this project. And this is a project that's being carried on in collaboration with Thomas Sauvage, who's a graduate student in the botany department. Okay, let's have a closer look at the Alvales using this same marker, the nuclear LSU. Now, what we wanted to do by using the Bishop Museum material was be able to put names on things with the assumption that the Bishop Museum material is expertly identified. Okay, now everyone's smiling, I can see that, but what I did was actually go and select sheets that were identified by taxonomic experts in this group. Okay, so I have a lot of faith that they truly believe and they understand that this is, you know, what they think these species are. There is absolutely no blame attached to this. All I'm doing is illustrating how much systematic revision there really is left to do in these groups. You can see right here at the top that Alva rigida and Alva fasciata seem to be the same thing, what we're calling these species here in the Hawaiian Islands. I'll show you with a second marker in just a few minutes that we see the exact same thing with another marker, so I think that's a good trend. Another thing is that the genus Enteromorpha is really, it's been sunk into Alva again, but I'm retaining these two names separately so that you can understand the difference between the flat blade-like forms, which are the Alvas, and the tube-like forms, which are the Enteromorphas. But even the Enteromorphas, there's a lot of confusion, I think, in what's going on here. For example, we can see down here we have two identical sequences from Bishop Museum material, Enteromorpha flexuosa, Enteromorpha paradoxa. We also have a flexuosa sample up here. Okay, so just a couple of small examples of the work that's left to be done. So more samples needed. We'd like to continue on with this group specifically. I think it's a great group to focus in on, and they're very amenable to molecular work. The fourth marker that we looked at was the nuclear ITS region. And you can see much the same trend that we saw for the LSU region, as you might expect. These are members of the same cistron. Okay, so again, ulva rigida, ulva fasciata, again, they seem to be the same thing, at least what we're calling here. Ulva reticulata, this is a really easily identifiable species. It's full of holes, has a net-like morphology. If you take these sequence here and you blast it on GenBank, we can do that because there's a lot of ITS sequence for ulva on GenBank, it comes up with a different name entirely. This is ulva onii, or it's the closest match to ulva onii, which is from Japan. Now, if I looked at the morphology of this, and they're very different, so I don't think what we have here, ulva reticulata, is ulva onii, but I think it's probably a close relative of it, and it may not be true ulva reticulata. It may be an undescribed species. And again, we see the same issues with the Enteromorpha group as we did with the last marker. So again, this is a group we're really interested in and would like to work more on. So just to summarize, the initial DNA barcoding approach was a little too simplistic for the complexity of this flora that we have and the state of the systematics that we have right now with this group. We encountered some problems with amplification and sequencing for at least some of the groups for all of the markers. But in the end, we are able to recommend these two markers as the ones that we'll be continuing on with and expanding this project with. So the plastid, the universally amplifying marker, we're going to continue to use. You can see it worked for most of the groups, except the Clinophoreales. And the nuclear LSU region, we're also going to be continuing on with that. Again, the Dasycladales were not so successful, but hopefully we can get at them with the other marker. But we also have this group here, the Alvales. This was an extremely wonderful group to work with. It's the only one that seemed to amplify with everything that we did. Easy to get the DNA out of this group. They don't seem to be full of secondary metabolites. It's not calcified. It doesn't have a lot of secondary substances that really interfere with molecular protocols. So I really want to continue on with this, expand this project, and choose this as an example group to really hone in on the systematic revision as a place to start with the green algal flora. Major conclusions. Systematics of the Hawaiian green algal flora is far from settled. Molecular markers can aid in the identification of taxa requiring systematic attention, so that's one of the benefits of doing an analysis like this, even if everything isn't completely settled. But at the end of the day, our original aim was to be able to provide species identifications using this sort of technique. And species identification with molecular markers won't be any better than with morphological characters until systematic revision for each of these groups is complete. But I want to put in this caveat here that we can still demonstrate allegiance with known clades if we have a relatively well-populated molecular framework, and then we go back and essentially take a reverse taxonomy approach and start to pick out these groups one at a time and work through their systematics in a lot of detail. Recommendations for markers: all samples will be using the UPA marker and the nuclear LSU. When we hone in on the Alvales, we're also going to be using the nuclear ITS, and that's because this is the region that has been used for the systematics of this group in the past. There's a lot of GenBank data available for comparison. Okay, so we're expanding our work on the Alvales. We'd like to include, not just 30 or 40, but hundreds of these in our networks. We'll be mapping on our morphological characters and going back to look at these specimens, these vouchers, in a lot more detail. And the Hawaiian samples of Caulerpa are also being investigated in a lot more detail in collaboration with Thomas Sauvage. Okay, so I'd like to end off by thanking the Hawaii Coral Reef Initiative for their funding of this project. I'd like to thank all of the people who contributed specimens, collecting time and effort on this project, and a lot of the folks at DAR who really helped us out. And thank you.